A-Level Biology 9700, Paper 4, Variant 2, October-November 2025. And in this video, we are going to discuss question number 3 and 4. So, I start with the question number 3. The lac operon is present in the genome of the bacterium Escherichia coli. When glucose is not available, the presence of lactose in the extracellular environment leads to the expression of the genes of the lac operon. This leads to an increase in the uptake and metabolism of lactose. When glucose and lactose are available in the extracellular medium, lactose is prevented from entering the bacterial cell. Figure 3.1 shows what happens when glucose enters a bacterial cell. So glucose enters the bacterial cell using a transport protein at A, and B, capital B is an enzyme found in the cytoplasm of the bacterial cell, and B catalyzes the phosphorylation of glucose to produce glucose 6-phosphate. So here you can easily observe the figure 3.1. Now, question is, with reference to figure 3.1, suggest and explain how the presence of glucose in the extracellular environment prevents lactose entering the bacterial cell. So we have to suggest and then we have to explain it as well. So that glucose transport and phosphorylation, how do the glucose transport and phosphorylation occur? So you know that the glucose enters the cell via transport protein, as it has been shown in the figure. So once inside, enzyme B transfer a phosphate group, uh, dephosphorylation of to glucose converts it into glucose 6-phosphate. So dephosphorylation of enzyme B occurs when glucose is being actively transported. Enzyme B remains in its dephosphorylated state because it is constantly giving its phosphate to glucose. So the diagram shows that the dephosphorylated form of enzyme B interact directly with the lactose permease, the transport protein for lactose. When this interaction causes a conformational change in lactose permease or physically blocks it, so preventing lactose from moving from the extracellular environment into the cytoplasm. So I would like to write all this information which I have explained to you, that it happens in this way. So I would like to write it for 3 marks, that is the glucose enters. Glucose enters the cell via transport protein A, then enzyme B transfer phosphate group. To glucose, converting it glucose into glucose 6-phosphate. So then when glucose is being being actively transported enzyme B remain in its dephosphorylated state. Which directly interacts with the lactose permease this causes a conformational change conformational change conformational change in lactose permease preventing lactose from moving from the extracellular environment into the cytoplasm. So it was the part one of this question. Now the part two of this question is suggest the advantages for the bacterial cell in preventing the entry of lactose when glucose is present in the extracellular environment. So the advantages can be as uh, more energy will be available, less energy will be, you can say, uh, wasted, and the amino acids will be, you can say, they will also not be wasted. So I would like to write that the glucose, that is a monosaccharide and a respiratory substrate, so that can enter glycolysis directly. So when the metabolism of glucose is taken place, the fewer enzymes at less energy break down lactose. So in this way, the it will prevent the entry of lactose into the glucose is extracellular. So glucose monosaccharide that can enter directly, directly requires less energy and fewer enzymes. The cell avoids wasting ATP and amino acids synthesizing enzyme. Enzymes and which are only necessary when lactose is the primary source. For example, we can write its name beta galactosidase, name of that enzyme. So next part of this question is the glucose 6-phosphate in the cytoplasm of the bacterial cell is used to produce fructose 1,6 biphosphate. Fructose 1,6 biphosphate is used in glycolysis. Outline the events that occur in glycolysis following the production of fructose 1,6 phosphate. So we have to write down the different steps which can take place in glycolysis for the production of fructose 1,6 phosphate. You know that fructose 1,6 biphosphate is formed, the energy conserving stage of glycolysis begins. So the carbon 6 carbon fructose 1,6 biphosphate is split into two molecules of 3 carbon intermediates known as triose phosphates. So each triose phosphate molecule is oxidized by losing hydrogen atom. So these hydrogens are accepted by the coenzyme NAD, reducing it to reduced NAD. Then the energy released from the oxidation of triose phosphate is used to synthesize ATP. Since there are two TP triose phosphate molecules, a total of 4 ATP molecules are produced in this stage. So the final 3 carbon product of these reactions is pyruvate, which then moves to the next reaction, which is the link reaction. 6 carbon fructose, fructose, 1,6 biphosphate, splits into two molecules, three carbon intermediates known as triose phosphate. Each triose phosphate is oxidized by losing one hydrogen atoms, which are accepted by the coenzymes NAD, reducing it to NADH. The energy released from the oxidation of triose phosphate is used to synthesize ATP. The final three carbon product of these reactions is pyruvate, which then moves to the link reaction. Link reaction is the second stage of respiration. You know that. So it was all about the part 3 of this question. Then move towards the part B. So in part B, you can see E. coli is present in the gut of young mammals. The diet of young mammals contains lactose, which is present in the milk they consume. Scientists carried out an experiment using E. coli with a lag operon and E. coli without a lag operon, with and without. So the experiment was carried out to determine whether the presence of a lag operon gives E. coli a selective advantage. Equal numbers of lag E. coli and lag E minus E. coli were given to 8 mice to allow the beta to colonize the gut. One day, 0 to 50% of the E. coli of the gut of each mouse were lag E. coli. 4 mice were fed a standard diet that include lactose. 4 mice were fed a standard diet without lactose. The percentage of lag E. coli that were in the gut of each mouse were determined every day for 6 days. Now you can compare these two curves with lactose and without lactose. So you can see it starts from the percentage or 50%, and then with lactose has been shown a curve above, and the without lactose has been shown a curve which is little bit curved, not curved, little bit curved. So not showing too much data or after many days. But lactose, the percentage of E. coli is more as comparison to without lactose. So question is, with reference to figure 3.2, describe the effect of the different diets on the percentage of E. coli that are lag plus. So here you can see, according to the graph, as I told, in both diets, the percentage of lag plus E. coli increases over the 6 day period, but the increase in other is not faster without faster as comparison to diet with lactose. So on the lactose diet, the percentage is high on the 0 to 6 day as comparison to the other without no lactose diet. So I would like to write the answer in this way, that in both diets, the percentage of lag positive E. coli increases over the 6 day period. And the increase is significantly greater and faster in the diet with lactose. On the lactose diet, on the lactose diet, the percentage rises from 50% to 86% by day 6. Whereas on the no lactose diet, the percentage only rises from 50% to 57% between day 0 to day 3. 58% is 0, day 6. So it was about part 1 of the B, section B. Now the part 2 of the section B is the scientists concluded the lag coli had a selective advantage when colonizing the gut, but only in the presence of lactose. Suggest why the presence of lactose caused lag E. coli to have a selective advantage. So I would like to say that the lag plus bacteria possesses the lag operon, so which allow to produce the enzymes as in the last question I had told you beta galactosidase, so which is necessary to digest lactose. So in an environment where lactose is present, bacteria can use it as a respiratory substrate, and whereas the lac minus bacteria cannot do that. So because they have access to more energy, so glucose lac plus bacteria can grow and divide faster than lac minus bacteria. So over time, the lac plus bacteria out compete the lac bacteria, increasing their frequency in the population. So it was the suggestion that why the presence of lactose caused E. coli to have a selective advantage. So I will start it because the question is of only 2 marks, but I have given you more detail. So in an environment where lactose is present, lac positive bacteria use it as respiratory substrate, whereas the other cannot. C cannot because they have excess, or you can see the lac plus can have more energy, have excess more energy, so can grow and divide faster. Grow and divide faster than lactose. Over time, the lag bacteria outcompete, increasing their frequency in the population. Because in this case, we have to look for the selection pressure. So selection pressure was on that side, that whether they are using it or not. So the selective advantage was that which they have got, and they increase their frequency and the number. Question number 4, the evolution in antibiotic resistance in bacteria has occurred as a result of natural selection. Name 2 ways in which a bacterium can become resistant to an antibiotic. So we have to write the 2 ways. So according to this, I would like to write that the one is the mutation. Mutation, what is mutation? Is a random change or the sudden change in the DNA of an organism, is in its number or type, or you can say structure of the DNA. And then the other, I would like to say, they can acquire resistance genes from other bacteria, most commonly through the process of conjugation or transformation or transduction. So the two are sudden change in number and structure of DNA. So this is the mutation, and other is the from other bacteria by conjugation. In conjugation, two bacteria come close to each other. So in this way, they can acquire the resistance from one another. Next question, the World Health Organization regularly analyzes bacterial DNA sequence data. Suggest one way in which this contributes to solving the problem of antibiotic resistance in bacteria. So for this purpose, the World Health Organization, uh, they can perform the following as first of all, identification of specific resistant genes, uh, and then determination which antibiotic will or will not be effective against a particular strain. Then track the spread, how the resistance of these strains geographically or between different populations, to implement better, you can say, the containment strategies. So we can identify new targets as for drug development by observing how bacterial genomes are evolving. So these are the, you can write on the one step in this, right? First of all, determine which antibiotics will or not or will not be effective against a particular stone. Track the spread of resistant stones. And then identify nutrients. Well, the last part of question number 4C is some infectious bacterial diseases are treated with the antibiotic streptomycin. So, if a person does not finish the prescribed course of streptomycin, bacteria are more likely to become resistant to the antibiotic. Explained why a streptomycin strain of bacteria is more developed as a result of natural selection when a person does not complete the prescribed course of antibiotics. So here we have to, you can say the process follows the principles of natural selection when a course of antibiotic is not finished. So we can see that the variations exist within the bacterial populations. Some individuals may have a degree of resistance due to random mutations. The antibiotic acts as a selective pressure, so when the course is stopped earlier, the concentration of the drug is high enough to kill the more susceptible bacteria, but not high enough to kill those with uh, partial resistance. So the bacteria with resistant alleles survive in complete treatment. So these surviving resistant bacteria multiply rapidly, causing they no longer have competition from susceptible bacteria. So they pass the resistance alleles to their offsprings. So over time, the frequency of the resistant alleles increases, leading to a population that is predominantly resistant to streptomycin. So this is of 3 marks. So briefly, we have to explain this. Variations exists within the bacterial populations. Some of them show resistance due to mutation. The antibiotic act as selection pressure, which kills the other bacteria but not the muted one. These surviving bacteria divide and multiply in number and make a population. and pass on the resistant allele to their offsprings. Over time, the frequency of the resistant alleles increases. So it was all about question number 3 and question number 4. So please like, share, and subscribe my channel for more videos on this paper and other papers. Thanks for watching.